Welcome back to the lecture series in role of nanomaterials in agriculture. So, we are into the animal production section. So, the question where I ended my previous lecture was, could antioxidant therapy improve endurance of skeletal muscle? Because I mentioned you that during exercise it generates reactive oxygen species, and those ROS are essential for generation of contractile forces, but during intense exercise there is a large quantity of reactive oxygen species which is generated. Unless otherwise your system is well equipped with fully functional superoxide dismutase, glutathione peroxidase or catalase, if they are not well equipped with it, then you may suffer from contractile dysfunction, muscle weakness and fatigue. Okay. So, according to the current understanding of skeletal muscle physiology, ROS or free radical at low and physiological level is essential for normal force production. Okay. Whereas at a higher level of ROS causes contractile dysfunction resulting in muscle weakness and fatigue. Such ROS mediated skeletal muscle tissue damage often occurs during intense physical exercise and is termed as, there is a term used for it, exercise induced oxidative stress. Exercise induced oxidative stress. Thus, the level of ROS forms a pivot which regulates the exercise redox biology of the skeletal muscle. Exercise redox biology of skeletal muscle. Now, one of the option is supplementing the diet with antioxidant like vitamin E. Okay. Supplementing diet with antioxidants like vitamin E, and these attempts has been attempted to reduce the ROS load or reactive oxygen species load in the skeletal muscle, thus reducing the ROS mediated oxidative damage. Single large scale intervention experiment using high doses of antioxidant offered disappointing result. So, what has been observed that the studies where such animals have been supplemented with heavy doses of antioxidants, and which are mostly, if you look at vitamin E, the structure of vitamin E, these are huge molecules. Okay. The results were disappointing. Now, that is very critical, and that needs a very sane thinking. Now, I will again take you back to one of the very previous classes when I talked to you naturally occurring antioxidant molecule, if you remember it. Remember? So, this naturally occurring antioxidant molecule, these are huge molecules, something like this. And I told you these are used in food additives in the industry, but these huge molecules, when you are putting into the system, in order to counter the ROS, they may have their own side effects too, because body is not used to consume these molecules in huge amount. Like if you take a lot of vitamins, it is not going to help you really. I mean, you may not even need vitamin because body may function with a very trace amount of vitamin. You may need vitamins. What I mean to say, you may not need vitamin supplementation really at times. We always get, oh, take as much as possible. That is a very wrong concept. That does not work because body has its own way to conserve them. Now, if you are supplementing the diet with lot of this, say for example, in this case the whole trial failed of vitamin E as an antioxidant, it completely failed. It did not work. because there are issues with such kind of trials. So, what I was trying to highlight is that just because we believe this is going to work, will work, it does not happen like that. And that was the reason why I highlighted this part. When there are these kind of huge molecules like this, they may come with their whole series of side effects and it may not work. So, we need an optimal quantity. So, this is the area where nano may come pretty handy, where the quantity is extremely low, some delta quantity we are talking about, and the effect is very profound. And when we talk about something like cerium, which is needed in extremely small quantity going by the reaction, which is having a pseudo infinite half life, then the storyline completely changes. So, getting back, what are the things which are observed was that single large scale intervention experiments using high doses of antioxidant offered disappointing results, that thus antioxidant nutrition studies as a therapeutic approach remained inconclusive. Till this date, it is one of the most inconclusive study that whether antioxidant therapy is good for animal production or not, or at least increasing their skeletal muscle endurance and all other stuff. Is it possible? And it is very, very inconclusive. But then at that time there was one study which was conducted using cerium oxide nanoparticle instead of vitamin E. This failed. Okay. Could we use CO2, C2O3, sorry, could we use this redox couple in a skeletal muscle? And the whole idea was, the key question is how cerium oxide exhibits such myriad effect, and we have already talked about it. Okay. So, now here this one study showed a very interesting result. It has been introduced into the rat by intramuscular injection. Okay. So, basically this whole trial was done on rats. Something like this. Okay. It has been introduced into the rat by intramuscular injection, and the skeletal muscle endurance has been evaluated. Intramuscular injection of cerium oxide concurrent with exercise. You have to do the exercise, you have to make the rat go through the exercise. It enhances, there are 2-3 things which enhances. It enhances the muscle mass. There is a huge enhancement in muscle mass. There is an increase in glycogen. There was an increase in ATP content. One second. Okay. And type 1 fiber ratio. There is an increase in. So, these are the different fiber ratios of the skeletal muscle. Thus resulting in significantly higher muscle endurance. There is a higher muscle endurance. So, electron microscopic studies confirm the presence of cerium oxide nanoparticle in the vicinity of muscle mitochondria, the powerhouse of the cell. So, there were presence of the cerium oxide, and what is the significance will come about it very soon. Thus, there was an increase in the number and the size of the muscle mitochondria in the serum oxide nanoparticle treated muscle following exercise as compared to the untreated group with only exercise muscle. And apart from it, the quantitative proteomics data and subsequent biological network analysis studies identified higher level of oxidative phosphorylation, the major energy generation reaction of the body, the biological system, TCA cycle output, tricarboxylic acid cycle output, okay, and glycolysis, breaking down of the glycogen, lysis, okay, glycolysis, okay, glycolysis, okay, and this was further associated with a significant increase in the mitochondrial respiratory capacity and muscle contraction, primarily due to the higher level of electron transport chain proteins like, so all the electron transport chains which are proteins which are involved are NDUFA9, SDHA, ATP5B, ATP5D, okay, and was all these were evaluated by ICP-MS data, and which revealed clearance of the particles after 90 days without exhibiting any inflammatory or adverse effect on the health of the experimental animal. Thus, a higher physiological endurance of serum oxide nanoparticle supplemented exercise muscle opens a new avenue for skeletal muscle therapeutics, space and sports medicine. So, what if you look at the data very carefully, it results in increase in muscle mass. In other words, what you are countering is that pseudo-infinite half-life is countering the ROS damage. By countering the ROS damage, you are ensuring there is an increase in the muscle mass. Further, if there is an increase in muscle mass, there will be more glycogen. Those rich carbohydrates which are present there, which are ensuring that it could generate lot more energy. Now, if we look at the mitochondrial chain, electron transport chain, there is an increase in electron transport protein concentration. In other words, the efficiency of the mitochondria, which is the powerhouse of the cell. So, the efficiency of the mitochondria is increased because there is more oxidative phosphorylation, there is more glycolysis, there is more TCA cycle output. So, instead of using heavy doses of, where the study failed, if you look at it, in case of vitamin E, instead of using heavy doses of vitamin E, if you use a very trace amount of CO2, you may get totally dramatic results in a skeletal muscle. This is a very interesting finding in terms of increasing animal production. This study could be translated in poultry, where your meat production is extremely important. Okay, one area where for meat, similar is for beef, similar is the situation for sports animal, similar is in space medicine, because if you look at space, there is huge amount of muscle loss, especially the skeletal, mostly the skeletal muscle loss. If you see the data from NASA and other space agencies in the world, ISRO, NASA, and all other agencies, they will tell you, or the Russian agencies, French agencies, Chinese agencies, if you look at them, you will see once they come back, because of the no gravity, there is huge amount of skeletal muscle loss. How we can counter it? You need something in a very trace amount, and which does wonder. And this particular 5-atom or 3-atom inorganic molecule is doing wonders. I mean, the data is in front of you with the trace amounts where the biggest trial failed. There is an increase in muscle mass, which is very, very critical for the meat industry. Nutritionally, this is higher glycogen concentration. So the ATP content is increasing. So it means it has more energetically more robust. There is a type 1 fiber ratio which has gone up. This is very critical. Apart from it, you are breaking down more of the glycogen by glycolysis. And of course, this part, in future people will be studying how you can increase the percentage or concentration of electron transport chain proteins. So in other words, your electron transport efficiency in the mitochondria is increased. So these kind of studies are laying the foundation stone of application of nanomaterials in animal production, and could open up a altogether new avenue to explore nanomaterial in a very trace amounts. Of course, one has to be, and some of these studies have clearly seen within 50 days it is getting cleared up from the body, and slowly, because you are using a very trace amount, and even if it kind of these trace amount remains in the bone, it's not going to affect you, because in the bones along with calcium they may form complexes and they will remain there without really harming your system. So some of these studies in decades to come will lay a firm footing into the animal production world. And a small story which started back in UCF, University of Central Florida, Orlando, somewhere around 2003, 2004, is now almost there, where it is a technology, a small discovery, and if you look at it, all its applications, neuroprotective for spinal cord repair, cardioprotective for the heart health, radioprotective for cancer therapy, photoreceptor for retinal damage, H2O2 sensor for application, and now adding up to that, skeletal muscle endurance studies, and maybe sports medicine and space medicine, sports and space medicine. Apart from it, it found application in countering hyperbaric, means low pressure, hypoxia, low oxygen, survival. So in other words, animals production in places like Ladakh, Leh, higher reaches of Arunachal Pradesh, where you always have lower oxygen tension, these kind of administering this kind of molecules could make tremendous difference in the animal production. So, I will close in here, and I will provide you all the reading materials, the papers which have been produced in this area, that will kind of help you to appreciate the field, how it has emerged out from a small place, and today it is one of the very important nanomaterial which is finding wide range of applications from human to the animal world. Thank you.